For decades, psychiatric medicine operated on a chemical deficit model. If a patient was depressed, they lacked serotonin. If they were anxious, they lacked GABA. Modern neuroscience identifies mental illness as a physical degradation of the brain's architecture. Conditions like major depression, PTSD, and chronic stress are architectural failures. Chronic stress causes the physical protrusions on neurons, the dendritic spines, to atrophy. Synaptic connections wither. The brain becomes physically less connected and less capable of forming new cognitive patterns. Conventional antidepressants operate within this degraded architecture. An SSRI floods the synapse with serotonin, but the withered dendrites remain. The drug adjusts the chemistry inside a compromised structure, which explains why these treatments often take weeks to work and why symptoms frequently return when the dose stops. Psilocybin functions as a structural renovation agent. It physically rebuilds the neural architecture that stress and trauma have destroyed. In 2021, researchers established the baseline metric for this process. A single dose of psilocybin triggers a 10% increase in dendritic spine density in the frontal cortex. This structural change occurs within 24 hours. This finding reorients the objective of modern psychiatry. The goal shifts from the chronic daily management of symptoms to the acute induction of structural neuroplasticity, giving the brain the physical capacity to change. The structural evidence comes from a 2021 study by Linda Shao and colleagues at Yale. Using 2-photon microscopy, the research team tracked individual dendritic spines in living brain tissue, monitoring identical cortical neurons in mice before and after psilocybin administration. They focused their imaging on the medial frontal cortex. This region governs executive function, emotional regulation, and cognitive flexibility, the precise domains that become rigid during depression and trauma. This data shows the timeline of growth. By the 24-hour mark, neurons grew 10% more dendritic spines, physical scaffolding materializing overnight. The drug clears within hours, but structural changes persist. Checked 1 month later, the new spines remained fully anchored and robust. This localized cellular growth translates into behavioral shifts. In models of chronic stress, mice typically develop learned helplessness and anhedonia. Following psilocybin administration and the subsequent spine growth, those behavioral deficits reversed. Because the physical remodeling of the brain outlasts the pharmacological presence of the drug, we have a biological explanation for why a single session can lead to long-term psychiatric remission. This raises a precise mechanistic question. If standard serotonergic antidepressants do not force rapid structural expansion of the cell membrane, by what exact molecular pathway does psilocybin achieve it? The answer centers on brain-derived neurotropic factor, or BDNF. BDNF is the primary signaling molecule for neuronal growth and survival, the chemical fertilizer for the brain. In depressed and traumatized brains, upstream signaling failures suppress BDNF, preventing the brain from maintaining its synaptic connections. In 2023, research led by Alex Bhattacharya revealed a direct bypass. Instead of just working through serotonin receptors, the psilocybin molecule directly docks into the TrkB receptor. This biological trigger sends a signal down into the cell, activating the mTOR cascade. mTOR is the master regulator of cell growth. Once activated, it directs ribosomes to manufacture synaptic proteins. These physical building blocks push the cell membrane outward to form a new dendritic spine. The definitive proof of this sequence comes from rapamycin. When researchers administered rapamycin to block the mTOR pathway, the antidepressant behavioral effects of psilocybin were abolished, even though the psychedelic response remained unchanged. The clinical efficacy is strictly downstream of neuroplastic protein synthesis. The drug's ability to repair the brain relies on its capacity to physically manufacture new neural architecture, a process distinct from the subjective hallucination. This understanding of structural growth was catalyzed by David Olson's lab at UC Davis. In 2018, his team systematically quantified neurogenesis, spinogenesis, and synaptogenesis across different psychoactive compounds. This structural taxonomy chart categorizes a new drug class, psychoplastogens. Unlike traditional SSRIs, psychoplastogens rapidly force structural neuroplasticity. As the graph shows, psilocybin sits alongside ketamine, but exhibits an even greater magnitude of physical neural growth. The psychoplastogen model explains why single sessions yield lasting results. Once a physical scaffold is built, it functions independently of the original construction crew. The drug builds the architecture and then leaves. Furthermore, the Olson lab demonstrated that this neuroplastic growth can occur even at sub-hallucinogenic doses. The biological repair mechanism does not strictly depend on a massive perceptual shift. Isolating this structural mechanism opens a new frontier in drug development. It paves the way for scalable, non-hallucinogenic psychoplastogens, medicines that induce rapid neural growth and could be taken without the need for supervised clinical sessions. Clinical application views the acute psilocybin session as opening a biological window. The experience softens a rigid brain, pushing it into structural openness. This dual-axis chart maps the precise timeline of structural flux. The red line represents the drug concentration, which spikes and clears within 24 hours. But the blue shaded area represents the neuroplasticity window. The actual formation of new dendritic spines peaks at 72 hours and remains elevated for weeks. Prior to this window, conditions like trauma function as rigid neural attractor states that resist cognitive updating. This is where active psychotherapy becomes biologically necessary. The critical phase of therapeutic integration aligns strictly within this blue shaded area. Reprocessing traumatic memories during this window allows the patient to encode healthier cognitive patterns directly into the freshly generated neural scaffolding. Psilocybin administered without a structured therapeutic context consistently yields smaller, less durable effects. The drug builds the new structure, but without directed encoding, the brain defaults to familiar patterns. The biochemical cascade physically constructs the new neural architecture. The human therapeutic work determines what lasting behaviors, insights, and identities are built inside it.